Well, hello, my fellow pre-meds. In this video, we're going to talk about ribonucleic acid RNA. Specifically, we're going to be focusing on the genetic code and the process of transcription. I made the conscious decision of dividing this slide deck into two separate slide decks because biochemistry is complex as it is, so I like to keep these, as short as possible so as not to overwhelm you with a bunch of information in one video. So the second part of this video is going to cover translation and transcription regulation. Don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow and I would really appreciate it. So let's get started over here with the central dogma of molecular biology. So as you can see, the central dogma of biology is that the genetic information will flow from DNA to RNA to proteins. And that DNA is how our cells store this information. And then it translates it or transcribes it into RNA, which will be used in the synthesis of proteins at the ribosomes in the cytoplasm. And proteins are ultimately what carry out most cellular structure and function. Now, this is just a little diagram of that process over there, and throughout this slide deck and the next one as well, we're going to be discussing this entire process in detail. So let's start off with the one of the major categories of RNA, specifically messenger RNA. So messenger RNA is what's carrying, as we mentioned, genetic code from the nucleus, from DNA to protein, to the ribosomes to synthesize protein. All right, so mRNA is synthesized by way of RNA polymerase during transcription. mRNA contains codons that specify the amino acid sequence in the process of translation. So more on the codons in subsequent slides. In eukaryotes, mRNA is processed before leaving the nucleus. So we'll talk more about this process where you have mRNA after transcription, but it's not ready to be translated, okay? And then in eukaryotic cells, mRNA is typically monocistronic, whereas in prokaryotes, it's polycistronic. You can memorize that by P over here and P in polycistronic. What does this mean? This right here is an operon, a gene from a prokaryotic cell, and you can see how it's been transcribed into an mRNA strand, and each of these represents a protein that can be synthesized from this mRNA strand. And that's the idea behind a polycistronic mRNA molecule. If this were in a eukaryotic cell, each mRNA can only synthesize one protein, whereas here, the ribosome can start here, it can start here, or it can start here to synthesize protein F or protein C or protein A, etc. All right, now let's talk about transfer RNA or tRNA. So tRNA transports the amino acids to the ribosomes to be able to synthesize the polypeptide. Now, each tRNA contains a 3-letter nucleotide anticodon. So recall in mRNA, you have a 3-letter codon. In the case of tRNA, you're going to have a 3-letter anticodon that is complementary and running anti-parallel to, and we'll see exactly what that means in a bit, and it's running anti-parallel to the mRNA codons. So amino acids are attached to the 3' end because just like any other strand of nucleotides, there's always a 3' and a 5' end. So you can see in tRNA you have a 5' and a 3' end. The amino acid is going to be attached to the 3' end, and once that amino acid is attached, we then call it an activated or a charged tRNA, and we give it a different name where we call it aminoacyl tRNA. And this has high energy because that energy is going to be used in the synthesis or in the formation of the peptide bond. So you can see this reaction between an amino acid and a tRNA molecule, as you can see right here, is driven by the enzyme aminoacyl tRNA synthetase. So essentially you get the tRNA that doesn't have an amino acid, and you get an amino acid, in this case alanine, the specific amino acid that's associated with this anticodon. And then you also need ATP because it's going to be a high energy bond that you can use to form the peptide bond over there. So the outcome of this reaction is your aminoacyl tRNA. Notice here you have the D loop, the T loop, not very high yield on the MCAT. Really what you want to know for the MCAT is that you have this 5' end, which typically is phosphorylated, the 3' end where you have the amino acid, and then this anticodon region where you can see CGU in this case. All right, now let's move on to ribosomal RNA. So rRNA is a component of the ribosome. The ribosome is what ultimately will synthesize the protein, the formation of the peptide bond in translation. So it's formed of a mixture of proteins and a nucleotide sequence. Importantly, the rRNA in this case is what's carrying out the catalytic activity. So normally we think of enzymes as proteins. In this case, what's carrying the catalytic activity over here is the rRNA, and we call it a ribozyme. So in eukaryotes, rRNA is synthesized in the nucleolus. And then, as you can see right here, ribosomes contain a large and a small subunit. More on that when we talk about translation in the subsequent slide deck. And notice that this picture just brings it all together. So you have your large and small subunit, you have your mRNA strand, and then you have your tRNA, which is carrying the amino acids. All right, now let's talk about the genetic code and codons. Now that we've introduced the major types of RNA, let's talk about how they interact with one another. So, as we mentioned earlier, the genetic code is read in a 3 nucleotide sequence on RNA. So these three-letter nucleotide sequences are called codons. Each codon specifies a particular amino acid. And then you have, there are special codons, including AUG. AUG is the start codon, which will start the process of translation with an amino acid called methionine. More on that in our amino acid slide deck. And then there are other special codons, UAA, UGA, and UAG. These are stop codons, so they will terminate translation when you're done with the synthesis of the protein. A good way to memorize these is that UAA, you are annoying, UGA, you go away, and then UAG, you are gone. So that's how I memorize them, because they do show up on the MCAT, so it's important to memorize them. And then tRNA anticodons will be complementary to these. So in the case of UAA, for example, it would be UUA. So it's just complementary over there, and it's running antiparallel. And then you have the genetic code is degenerate, meaning multiple codons can bring the same amino acid. And then you can see this once again over here, the idea that they're complementary and antiparallel. You can see this one's running 5' to 3', so 5' UGC. Complementary to that is going to be 3' ACG. All right, now this is the codon table. You obviously don't need to memorize this, but you should be able to use one if they provide it to you on the MCAT. So you can see this is the first letter, the second letter, so you'll go U, and then the third letter will be whatever is over here. And sometimes there'll be like different variations of this table, but they're all pretty much read in the same way. And then you can see like UUU would be phenylalanine, and so on. All right, so now let's talk about mutations. And the first thing I wanted to talk about is the idea of this degeneracy to the genetic code that we introduced previously. So essentially, multiple codons can encode the same amino acid. And this is by design, because you want sort of that wobble position where if you had a mutation at one specific site, hopefully it won't, like you change one nucleotide, hopefully you'd be able to get the same exact amino acid, and that's actually what happens. So if you take leucine as an example over here, let's go back to our codon table, you can see that leucine can be encoded by CUU, CUC, CUA, and CUG. Now notice the first two letters are the same thing, are the exact same thing over there. And that's because just in case you have a mutation in that third letter, you'll still get leucine and you won't disrupt the overall structure of the protein. Now the we're going to talk about two different mutations. You have point mutations and you have frame shift mutations, although there are different mutations within each of these two broad categories. I'll very briefly run over these and then show you examples, because I think really the examples will really hammer the points down. So you have silent mutations do not alter the amino acid sequence of the protein. So you're replacing a codon that gives you leucine for another codon that gives you leucine. Missense mutation substitutes one amino acid for a different amino acid. And then nonsense mutation creates a premature stop. So you get one of those stop codons such as UAA. So you just stop synthesizing the rest of the polypeptide. And then a frame shift mutation results in from the insertion or deletion of a nucleotide, which is going to throw off the entire nucleotide sequence, as we'll see in this slide. So let's start off with that most complex one, which is the frame shift mutation. So you can see right here how the normal sequence was A, UG, UCA, GAG, UGU. Now, we only deleted one of these nucleotides, specifically U. So you wind up with AUG, CAG, and you throw off the rest of the sequence. So you can see whatever happened before the deletion is still the same, or before the insertion is still the same. You still have methionine as that first amino acid. However, anything after the addition or deletion is going to be thrown off. So you have UCA, gives you serine, but now, because you got rid of that U, it's CAG, and you get glutamine. And notably, everything after that will be a different amino acid because you've thrown off the entire sequence. Now, an important point over here is, if you were to add multiples of 3 nucleotides, so say I added 3 nucleotides or 6, you're not going to throw off the entire frame, because what do you think is going to happen? Like if I had AUG and I inserted, for example, AAU, that next amino acid is going to be a new amino acid, okay, but then the rest is going to be the same exact thing. So UCA is still going to be within frame, GAG, and so on. So it's really, if you add or delete 1 or 2 nucleotides, that's when you're going to throw off the entire frame. Now let's get back to the point mutations. So silent mutations, as you can see right here, you replaced UCA, you still have the UC part, and then you just replaced A for G, and turns out both of these codons will give you serine, so it's the same exact protein, same exact amino acids. Whereas when you're looking at the missense mutation, you replace the middle letter over here. This is usually not associated with the wobble position, so you don't get the same amino acid. So you go from GAG to GUG. GAG was glutamic acid, now you have valine. Now notice that glutamic acid is a hydrophilic charged amino acid, whereas valine is a hydrophobic amino acid. 2 completely different amino acids, so it's going to throw off the entire structure of the final 3D protein. And this is actually what happens in sickle cell disease. You replace glutamic acid for valine. Now, an important point over here is that a missense mutation will not always result in this loss of function of the protein. And that's because if you were to replace, say, glutamic acid with aspartic acid, they're both hydrophilic, polar, acidic amino acids, and therefore they have pretty much the same role within the protein. So you can get slight deviations from the normal structure, but carry out the same function. Likewise, if you were to replace valine with another hydrophobic amino acid, you may not throw off the entire structure of the protein. Now, when it comes to a nonsense mutation over here, essentially what you're doing, you can see right here, you have GAG, and now you have, instead of that first G over there, you have a U. So now it's UGA. So essentially what you did over here is you've introduced a stop codon. And what's going to happen here is you're going to have premature stop of the synthesis of the protein, which is obviously going to throw off the entire structure of the protein. Now, if you get missense and nonsense, they're very similar in terms of the naming. The way I memorize them, to not get them confused, is imagine you have like some construction work, or I don't know, like landscaping you're doing, and then one day you come home and you see the person who was supposed to be doing the job is just not there, left all the tools, all the supplies, didn't finish up the job. Like that's complete nonsense. That's BS, okay? However, if you came one day and you saw that instead of the person who you hired was there, someone else, maybe their coworker was there instead, well, that's not complete nonsense, because that person may be able to carry out the same exact job, or maybe even better than the original person. So that's just like a missense, you know. It's not complete nonsense. Okay, that's just the way I memorized it. All right, so now let's talk about transcription. So transcription is carried out by RNA polymerase, and the function there is to generate an RNA transcript from a DNA template. Now, how does RNA polymerase know where to start? Well, it's this promoter region that is going to bind to. It's not going to start at the promoter region, but it just gets it close to where it should start transcribing. And there are transcription factors that'll help it locate this promoter region. Also, it'll be able to identify the TATA region, which is part of the promoter. So TATA, you just have multiple T and A, so thymine and adenine. And that's sort of part of the promoter over there. Now, it says over here that it's -25. That's just the location. So the way we name the location of nucleotides on a gene is that anything that is before the transcription site or upstream of the transcription site is going to be assigned negative numbers. So it makes sense. The promoter region is before where you start transcribing, so it's going to be negative. And then anything downstream is going to be positive. And essentially, like +1 is going to be that first nucleotide that you're going to transcribe. There is no 0, so it goes from -1 to +1. Okay, and then RNA polymerase, like DNA polymerase, will read in a 3' to 5' fashion to synthesize the new strand in a 5' to 3' fashion. If you want to understand the underlying mechanism over there, check out my video on DNA replication. Importantly, unlike DNA polymerase, RNA polymerase does not need primers to start that process for it. So it can start the synthesis from scratch, whereas DNA polymerase needed primase, recall, to insert those first few building blocks, those first few nucleotides in order for it to continue its function. And now this is very important. So the initial RNA transcript, particularly in eukaryotes, that you get from this process is called a heterogeneous nuclear RNA, hnRNA, which then goes post-transcriptional processing to form the mature mRNA. More on this, it's extremely important, more on this in a few slides. So this is essentially the process of transcription. So you can see helicase will unwound that DNA, double-stranded DNA, and then RNA polymerase will locate the promoter and then start transcription. And then as you can see right here, so once you get your messenger RNA, you're not done. You still need some processing to do before it can exit and go to the cytoplasm in order to continue with translation. So we will revisit that slide in just a second because I do want to highlight a few more things over there. You want to know the different kinds of RNA polymerases. So when I talked about DNA polymerase, there's not much you need to know about the different types. It's not very high yield. But the RNA polymerases, you do want to know that RNA polymerase 1 synthesizes rRNA, which is the ribosomal RNA, in the nucleolus. RNA polymerase 2 synthesizes mRNA, or messenger RNA, and RNA polymerase 3 synthesizes tRNA and some rRNA, but it's more relevant that it synthesizes tRNA. And the way you can memorize this is that rRNA is all the way in the center in the nucleolus, so it's going to be RNA polymerase 1. mRNA 2 is going to be in the nucleus in the process of transcription and then post-transcriptional processing. So it's going to be RNA polymerase 2. So we're getting a little further from the center. So that's why it's number 2. And then finally, tRNAs are going to be located in the cytoplasm in the process of translation. So they are going to be synthesized by RNA polymerase 3. That's just how I memorized these. Now, what is the coding sense, the coding or the sense strand? This is important. It shows up on the MCAT. It's some important terminology. So the idea here is, and I want you to pay very close attention. So this right here is what we call the template strand. Essentially, RNA polymerase located the promoter and started to transcribe this RNA strand. Okay, so this is what we call the template strand. Now, the RNA strand is going to be complementary to the template strand, correct? What else is going to be complementary to this template strand? Well, the other DNA strand over here. This DNA strand, we call it the coding strand. The coding strand is essentially going to be identical to the transcript. Now, this isn't entirely true, and that's because there's one difference between RNA and DNA, notably that instead of having thymine, RNA has uracil. So how would this be different? Like, let's say this strand started off as 5' G, 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 A. Okay, what would you have on the complementary strand over here? Well, it would be 3' because it's antiparallel, so it would be 3' C, 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 and then the last one was A, so we would have T over here. Now, if you were to look at the RNA transcript, you would see that it is, again, 5' because it's identical to this one, so 5' C, 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 but complementary to that adenine, instead of having thymine, is going to have uracil. So essentially, it's going to be the exact same as the coding strand, with the exception of uracil being replaced in the place of thymine. Other terminology over here is that the template strand is called the antisense strand, whereas the coding strand is called the sense strand. Okay, so that's what I have written down here. All right, now I think this is the last topic we're going to discuss here, which is post-transcriptional processing. Pretty straightforward, but I think it's very high yield, so really try to memorize these. They'll be easy points on the MCAT. So you have at the 5' end, so after you've transcribed your messenger RNA, you have to do some processing. The first thing you're going to do is you're going to add that 5' cap. So the 5' cap is going to contain, as you can see right here, 3 3 phosphate groups. It's also going to have guanine in it, so sometimes you'll see like a 5' G cap because it contains the nitrogenous base guanine, and it's gonna attach at the 5' end of the mRNA. Now the 3' poly A tail, obviously on the other side, you're gonna have multiple adenine base pairs at the very end, or just bases, nitrogenous bases at the end, and these essentially protect it against degradation. So essentially, once you want to get rid of mRNA, if you want to degrade it after it has fulfilled its function, essentially you're looking for the messenger RNAs that lack that poly A tail. And you can think of the poly A tail as like a ticking time bomb. Like when it's first synthesized, it's going to be really long, and that signals that this should not be degraded. We're going to use this for translation. But once its function has been done, that poly A tail will start to shrink, and then eventually it's completely gone, and you can target the RNA, the messenger RNA for degradation. Now, here's where we're going to talk about introns and exons, and I'm going to tie it back to the idea of polycistronic and monocistronic, just so that you don't confuse these two very similar concepts. So you have an exon, an intron, an exon, an intron, an exon. Essentially what's going on over here is that for every messenger RNA or heteronuclear RNA that you've just transcribed, it has a mixture of exons and introns. Now, what are eventually going to be involved in the process of translation at the ribosome are these exons. The introns will be cut out, and they're going to be cut out by machinery called spliceosomes, essentially a mixture of RNAs and proteins. Now, the idea behind alternative splicing is how we go from, as humans, for example, having 20 or 30,000, these aren't exact numbers, but 20 or 30,000 genes, and then getting like upwards of 90,000 proteins from those 30,000 genes. How do we end up with that? So the way we end up with that is that this over here is a heteronuclear RNA being combined in multiple different ways. So as you can see right here, blue with black with red, the black exon with the blue exon, the red exon with the blue exon, the blue exon by itself. Okay, so you're getting multiple different mRNA transcripts from one heteronuclear RNA. So unlike when we talked about polycistronic versus monocistronic, recall in prokaryotes you have they're polycistronic because you can get multiple different proteins from the same messenger RNA. Here you can get multiple different messenger mRNAs from the same gene, but then once you've developed your final mature messenger RNA, from that you can only get one protein. But because we get multiple different mRNAs from the same gene, you can get multiple different proteins from the same gene. Notice here, you just have 3 exons and 2 introns. Very simple. But look at how many different combinations I've formed, and you can even form more than this. And that's how you go from like 20 to 30,000 genes in the human body to like upwards of 90,000 proteins. And those aren't exact numbers, okay? But you get the idea that you have many multiples more of proteins compared to genes in the human body. All right, now let's do these knowledge checks over here. So I usually just push through these, but if you have any questions, just reach out to me and I'll be glad to help out and explain these. So pause the video, try to answer this, but I'm going to push through. There it is. Here's our next question. All right, and finally, this question over here. Very high yield. Okay, so that concludes our slide deck on the part 1 of RNA and the genetic code. The next slide deck is going to talk about translation and transcriptional regulation, so definitely check that video out as well. If you have any questions, reach out to me. This is a dense topic. I mean, it feels like I say this every time because biochemistry is really a dense subject, so definitely utilize our Anki cards and access our slide decks to annotate if you feel like you need to annotate. And if you have any questions, be sure to reach out to me and go crush the MCAT. You got this. These videos, at least as of now, are sponsored by you. So if you're interested in supporting me to be able to continue to make these high quality MCAT playlists, you can support me either via Patreon or by buying me a coffee. Links in the description down below, as well as the links for everything else, including the link to our growing pre-med companion Discord community.